I'd like to share with you an application of the DILIsym model where we have used it to perform analyses on clinical biomarkers to determine the severity of liver injury. There are multiple applications, really, of DILIsym in the clinical development realm, and these applications range from the preclinical all the way to the clinical side, and really it's been designed for this purpose to assist in evaluating hepatotoxicity risk for compounds throughout the pipeline. In this particular presentation, we'll focus on using DILIsym along with measured clinical DILI biomarkers to determine the severity of injury. This approach has been used for multiple candidate therapeutics, and results from these projects have been included in communications with regulatory agencies. Before we describe the project itself, I'd like to provide a brief overview of the DILIsym modeling software. There are 4 species included in DILIsym: human, rat, mouse, and dog. And each of these species includes inter-patient or inter-animal variability. The cellular processes that are really essential to modeling mathematically drug-induced liver injury are included, and this diagram is kind of a summary of the primary submodels within DILIsym. You can see that there's a submodel of drug metabolism and distribution that enables then the prediction of parent and or metabolite that may interact with the various mechanistic pathways associated with hepatotoxicity, which include, for instance, the bile acid transporters, the generation of reactive metabolites, disturbances in reactive oxygen species, and or mitochondrial toxicity. These submodels interact in addition to lipotoxicity, and together they can disrupt the hepatocyte life cycle by promoting either apoptosis and or necrosis. There's also an interaction with the innate immune system to exacerbate or ameliorate injury. I should also point out that hepatocellular proliferation, particularly post-injury or regeneration, is included in DILIsym. It's also probably most important for this particular presentation to note that there is a submodel of biomarkers of hepatocellular injury included within DILIsym, and I'll explain this, I'll elaborate on that in a subsequent slide. While this diagram describes kind of in qualitative terms how these various biochemical aspects are interacting, we have used exemplar compounds, and a substantial number of them, to establish the quantitative interactions within and between each of these submodels. DILIsym is also an ordinary differential equation based model, really a compartment type modeling, and also includes a form to function connection that enables us to properly track mass balance despite the potential for losing hepatocytes due to predicted injury. So how do we then infer hepatocyte loss or estimate hepatocyte loss from clinical data and using DILIsym? And this comes back to our underlying ALT submodel by and large. So we'll start with this very simplistic representation. You can see there are the viable hepatocytes with ALT within them. And then there's a separate compartment for liver ALT, representing by and large ALT within, say, the sinusoidal space, ALT in the plasma, and then the ALT that has been cleared. So the release of ALT from hepatocytes is primarily driven by the rates of necrosis or apoptosis, and actually it's in the apoptosis area, it's really more captured by secondary necrosis. And then we can look at these ALT curves, and by knowing the quantitative relationships between cell death and ALT release, that we've established in assembling DILIsym, we can use it actually in reverse and infer the hepatocyte loss associated with a given dynamic curve of ALT. This is an iterative approach that ultimately leads to, by design, a correspondence between simulation results and measured data, such as is illustrated in this figure, where the red are the simulation results, the black are the measured data. And we first do that in the baseline human, but then we extend it and perform similar simulations in a wider range of simulated patients that we describe SimPops, and this includes inter-individual variability in various aspects of ALT profile, and this allows us to estimate then a range of hepatocyte loss for a given ALT profile. Taking that even one step further, we can also predict the distribution of patients within a predicted ALT range that may have a given amount of hepatocyte loss. We can also estimate the probability of severe liver injury, otherwise known as, say, Hy's law. And I should also point out that while most of this description is focused on ALT, we can also use additional biomarker data to confirm these predictions and even to indicate the mode of cell death, whether it's necrosis, apoptosis, or some combination of the two. A critical component of performing this particular analysis is understanding the dynamics or the kinetics of the liver enzyme profiles. And so various ALT profiles can have the same peak value, but very different kinetics. And there are 4 curves shown in this graph that show 4 distinct kinetic profiles. And if you perform the analysis as I just described and play it forward, what you see is then that the predicted ALT loss for each of these different curves is really quite different. 3-fold higher in the one with the greatest area under the curve versus the one with the least amount of area under the curve. And really what this highlights is that the detailed time course of the liver enzyme data is really important in enabling the prediction or the estimation of hepatocyte loss. So we've employed this technique in a project, a collaborative project with a company by the name of Cleveland BioLabs, and actually we've written it up and published it with the reference given at the bottom of this slide. This therapeutic Entolimod is one that reduces radiation mortality by 40%, and this satisfies the FDA animal rule for efficacy. That said, even though this type of therapeutic wasn't required to have efficacy demonstrations in humans, it was required to undergo safety studies, and in these safety studies, there were elevations in ALT and AST observed. And in fact, it threatened the ongoing development of this compound, and it was at this point that we collaborated to use DILIsym along with the clinical ALT and AST data to infer the amount of hepatocyte necrosis associated with Entolimod treatment in these studies. So we started with the aggregated ALT data for all the patients. Again, looking at this, for the most part, the ALT elevations were really quite modest, with a handful of patients with relatively high ALT excursions. Also, it's worth noting that ALT was back to baseline for most of the patients within about a week. We parsed out all that data and really focused in on 3 curves: the maximum, the 95th percentile, and the median response. We then iteratively performed simulations such that we were in agreement with the ALT data, and again, this is by design. And taking that a step further, once we had the ALT dynamics properly represented, we went back and determined the associated hepatocyte loss, and you can see in this figure at the bottom that it ranged from 0.3% in the median to 3.5% in the maximum curve. Again, this is all for a single kind of healthy volunteer median patient. In order to gain an estimate of kind of what would happen in a wider population, we used our simulated populations, or otherwise known as SimPops, that does include interpatient variability in various aspects of ALT, to predict then the range of hepatocyte loss for a given observed peak ALT range. And keep in mind, again, I'll just emphasize that these are only valid for time courses similar to those observed with Entolimod. And even for the maximum ALT patient that had an excursion, a peak ALT of about 1,000, really the hepatocyte loss was quite modest, ranging from say 2 to maybe 5% in this particular set of data. While this is a relatively modest degree of injury, with other therapeutics, certainly the injury can be more substantial, the peak ALT can be higher, the duration of the ALT excursion can be longer, hence the hepatocyte loss can be greater. And we've extended this analysis such that now we can, in addition to just determining a possible range of hepatocyte loss for a given ALT range, we can also determine two things. One is kind of the likelihood or rather the distribution of patients within a given ALT range that can be associated with a range of with hepatocyte loss. We can also determine the likelihood of achieving a Hy's law case, which is the indicator of serious liver injury for most regulatory agencies. We can determine the likelihood of that for a given peak ALT and dynamic profile of ALT. And this can be determined or expressed visually, such as this graph here on the left, or in tabular form. But ultimately, really what this enables decision makers to do is to determine just how near or far each case may have been to serious liver injury. And the associated risk-benefit analysis can be performed in the ongoing clinical development of a compound. In addition to, coming back to the Entolimod case study now, in addition to predicting the magnitude of hepatocyte loss, we were also able to predict the rate of recovery, the regeneration profile within our SynPops that also included variability in hepatocyte proliferation rates post-injury. And you can see the range of time that it took to come back to complete restoration took about 2 to 9 weeks throughout this SynPops, with the median being about 3 weeks. So to summarize, we were able to use DILIsym in a project where we were able to analyze clinical biomarkers in addition to simulation results to determine the severity of liver injury. This has been published and is available for further inspection at your leisure. And to summarize kind of the outcomes of the project, really DILIsym analyses indicated that hepatocyte losses were relatively modest at less than 5%, and that full regeneration and restoration of the liver was predicted to occur within about 2 months. Putting that into context, reviewing the literature for other similar types of injury support the conclusion that this magnitude of hepatocyte loss probably does not represent a serious health threat. And it's also worth noting that these simulation results and simulation analyses were submitted to the FDA in various communications in support of the safety of Entolimod. Not only have we performed or had this outcome with this project, but also with other therapeutics and other similar projects. And we continue to support these efforts and these types of analyses for the clinical developments situations such as this. So with that, I'd like to thank you and conclude.